Hi, everyone. Welcome back. Welcome to our webinar, co-hosted between lakeFS and Seqera, where we're going to host today CytoReason. And they're going to tell us about disease models as data products and how they ship reproducible disease models using Nextflow, Seqera, and lakeFS. To begin with a short agenda, first I'm going to tell you a bit of the housekeeping rules and about Goldcast. Then we're going to do a short introduction to Nextflow and Seqera Platform. Then our co-hosts from lakeFS are going to tell us about lakeFS, the control plane for AI-ready data. And then we're going to welcome Poches from CytoReason to stage. He's going to tell us about disease models as data products at CytoReason. And at the end, we'll have some time for moderated discussion and a live Q&A. In Goldcast, you have a Q&A button that you can use to ask questions. Now, to introduce our speakers today, we have Poches, Systems Architect from CytoReason, Ido Avneri, Chief Business Officer from lakeFS, and I'm Florian, and I'm a Senior Bioinformatics Engineer at Seqera. Now, short housekeeping rules at the beginning. Be great if you could all introduce yourself into the chat. Let us know who you are, where you're from, and what you're working on. Throughout the webinar, we will post poll questions that you can see on the right-hand side in Goldcast in the poll menu. It would be great if you could answer these to let us know what you're doing with these different products and how you're using them. And as I mentioned at the end, we will have a live Q&A where you can drop your questions throughout the webinar in the Q&A tab, and we will try to address as many questions as we can at the end of the webinar. Okay, now to introduce Nextflow and Seqera Platform for those of you that are not familiar. Nextflow has become, over time, the standard for reproducible and scalable analysis as a workflow orchestrator. And Nextflow really offers a great developer experience, right, with some of the features that we all familiar with and love. It allows you to do very fast prototyping of your workflows for data analysis. Parallelization is a key aspect in Nextflow and allows you to do your analysis in a very scalable manner, right? You can code a Nextflow pipeline on a small test data set that runs on your laptop. And then when you're ready to go, you can scale this out to hundreds of thousands of whole genome samples or any other type of data in the cloud or on your HPC. Containerization is really the core of reproducibility in Nextflow, right? It allows you to write software today, containerize it and run it with Nextflow and still get the same results in a year or 10 years, as long as you run it with the same version. And then portability, right? The ability that you can write Nextflow on your laptop, but then go to any cloud environment, any HPC provider, any executor that you want, and it will run the same on your computer and in any analysis. And Nextflow really allows you to write your pipeline run once and then run it anywhere. Because of these great features, Nextflow has really become the standard, right, in the industry. And we really see this when we look at adoption and the number of citations of papers that use Nextflow in the field. Nextflow is used by most of the top pharma companies worldwide, and the amount of Nextflow run every day keeps growing and growing, which is great to see, to make science more reproducible across the board. About 40% of modern bio workflows are run with Nextflow right now, and we see more than 3 million executions each month. Now, Seqera is really the trusted platform for enterprise-scale bioinformatics using Nextflow. Seqera builds on Nextflow and is the best place to run Nextflow, and it powers the science that a lot of our customers are running each day. Seqera not just runs Nextflow pipelines, however, it also connects to your data. It allows you to do interactive analysis using studios. It ensures that you have quality and security and offers secure access to your pipeline operations. With Seqera Co-Scientist, we also have tools for AI and ML-assisted development and doing work in those type of areas. So Seqera can really help power your organization and scale the Nextflow analysis that you're doing day to day. One of the components in Seqera is Data Explorer, which provides unified data access and management to your data operations. Now, this is really great because it allows seamless integration and interaction with your cloud storage without having to move data around. And this is one of the key components we're going to talk about today that our co-host lakeFS is going to show you about, how you can use lakeFS as a data backend within Seqera Platform and for your Nextflow pipelines. And lakeFS is going to tell you all the features they have, and CytoReason is going to talk about how they've adopted Seqera, Nextflow, and lakeFS to ship data disease models as data products. With that, I will hand it over to Ido from lakeFS to tell us all about lakeFS.
Thank you so much. Hi, everybody. I'm Ido from lakeFS, and here to speak with you today about lakeFS, which is the control plane for AI-ready data. We just have literally 3 slides over here to talk about it, and then we'll pass that along to Ron from CytoReason. So what we do is we close or bridge an infrastructure gap for AI/ML projects by providing a control plane for data, which means that we give you data that is more easily reachable, trackable, reproducible. You can work with it efficiently and within compliance. We have thousands of organizations that are using us, very impressive ones, if I may say so myself. You can see a lot of these logos here. And we find ourselves mostly in AI and ML environments that are multimodal, meaning that they have combination of structured, unstructured, semi-structured data sets. So this can be iceberg tables with MRI images, right? But it can be any format whatsoever, and we'll talk about this soon. And we fit within your existing infrastructure and help close this gap. The problems that we often help solve are things like data access issues, right? Which can be something like, how do I quickly get access to data? What data am I allowed to get access to, whether it's locally with data that's sitting remote, or maybe accessing the data on a GPU sitting somewhere, right? How do I work on the data in isolation? So if we have one data science that wants to change the data set without impacting someone else sitting right next to them, how do they do that in a easy way? Or quite opposite, if I'm changing the data set and I want to make sure that my team can access the same data set, the same data that I change, how do I make that easy for them, right? We do all this in a reproducible, easy way. So it's very easy, so a lot of our customers have the challenge of, how do I know what was the data set that was used to train every specific model? And moreover, how did we get there? Right? So what are the changes that happened until that data set became that way, including how can I make sure that the data that I'm looking at is actually high quality data, and if for some reason we got bad data gone into our environment, how can we recover from that? And all of that needs to be done, as many of us know here on this call, in a compliant way that you can easily trace everything that changed, who changed what, where. And we solve these solutions by basically being at the infrastructure level in between the object store and whatever agents, tools, or users are interacting with that object store. So the object store can be cloud object store like Azure, AWS, Google Cloud Storage, but it can also be on-premise object store, MinIO, DeliCS, NetApp Storage Grid. There's many others. And what we do is we provide on top of that object store a Git interface. So imagine that you can take your bucket or your collection of buckets and you can branch them, make changes in isolations, compare different commits, merge, run a pull request with a human in the middle for that merge. And that, again, can be done by either your AI agents that are making these changes in isolations, by your different tools, or just by users, data scientists that are working with the data. And that really provides this, taking these engineering best practices from code and providing them on data really solves all the problems that we looked at before. That's lakeFS very quickly, and I'll pass it along to Ron for talking about how in CytoReason they take advantage of these two solutions together.
Hi, everyone. So I'm Ron from CytoReason. I'm a system architect in CytoReason for the past 3 years. And we're going to talk about disease model as data products. And the first slide is trying to capture what we'll be talking about in this presentation, which is how the process of building a disease model became a software-grade process where every data artifact is a release product, which is version trackable with the full history and has its own release cycle, whether it's dataset specific or disease specific, which has automated data validations based on E2 data modeling, which I'll be talking about in this session, which supports fast transition from prototyping to production-grade analysis, which introduces modular workflows, whether it's for model enhancement or adding new evidence generation workflow, which I'll be also talking about in this session, and has dynamic infrastructure that supports customer environment requirements, like security and isolation, and can also reduce cloud costs. And most importantly, I will talk about how we did all that with the support of Seqera, Nextflow, and lakeFS. Okay, so first of all, I'd like to introduce CytoReason. So before we get into the architecture, I will explain what CytoReason actually is and what we do. I want to emphasize that CytoReason first is a technology company. We are not a pharmaceutical company, and we are not a drug development company ourselves. And at the core of our technology, we have our disease models, as you can see. These are computational representations of human disease, and they are generated by a complex system that we call the model engine. The model engine is really the main focus of my session today, and I'll go much deeper into how it works and how we are evolving its architecture. As you can see, our scientists use the model engine to build the continuously improve these disease models, but generating the models is only one part of the story. The second major part of our technology is the CytoReason platform, which enables our customers to consume, explore, and work with those disease models. And as you can see here, we provide three main interfaces for doing that. The first is the computational environment. This is a cloud-based development environment where computational users such as bioinformaticians and data scientists can work with the familiar IDs such as VS Code or Cursor. Using the CytoReason SDK, they can write their own code and run their own computations directly on top of the disease model data. The second interface is our graphical user interface. This is designed primarily for biologists and other users who want to explore disease biology interactively through visualization and graphical dashboards without necessarily having to write code. And the third and the newest interface is Lina. Lina is our research agent. Lina provides a conversational chat-based interface for working with disease models. But I have to say it is more than just a chatbot. It also acts as a coding agent. It can use CytoReason SDK to generate and execute computational analysis against the disease model data and run those computations within a sandbox environment. For the rest of this session, I will be less talking about the platform, and I'm going to focus mainly on how we build these disease models and how we are evolving the model engine to treat them as version reproducible data products. Okay. So to understand the architecture, we first need to understand how a disease model is represented. And at CytoReason, disease models are represented as knowledge graphs. I guess most of you are familiar with that term. Now, when I say knowledge graph, I don't necessarily mean a graph database. I simply mean a network of connected entities. Most of these entities are biological entities, such as genes, cell types, proteins, and pathways, but they can also be clinical entities, such as disease, tissues, or clinical conditions. The connections between those entities are what we call evidences. And evidence is a quantified relationship between two entities, produced by at least two entities, produced by one of our analysis. To represent these evidences and the connections to entities, we used a standardized format called E2. As you can guess, this obviously stands for evidence and entities. Each evidence type, for example, differential expression, has its own predefined schema. The analysis that produce these evidences are called evidence generators. Each evidence generator can consume E2 data as input and publish E2 data as output. For example, if you have an evidence generator that might perform differential expression analysis and produce evidence that a gene is significantly differentially expressed under a disease condition. These analyses can be completely different internally, but they all speak the same standardized language, which is E2. That standardization is important because it allows us to replace, improve, or completely rewrite an analysis without affecting the rest of the platform. And once we stop thinking about the disease model as the output of one giant pipeline, and instead think of it as a versioned collection of standardized evidences, it becomes a true data product. It's versioned, it's reproducible, and it's composable and independently evolvable. That idea is really I would say, the foundation for everything I'll show in the rest of this presentation. Okay, so moving on to the model engine. So the model engine is basically split into two main parts: the search and dataset ingest and the model factory. Starting with the dataset search and ingest, the idea on the left side, we see the dataset search and ingest. The idea is that we have a process for processing, validating, and standardizing datasets, which we handle within lakeFS as standardized version data artifacts. This process runs regardless of having to generate a disease model, which is on the right side. And eventually, at the end of this process, we have a standardized datasets, sample metadata, and processed molecular data, which are aligned with each other. And by doing that, we enrich the CytoReason's dataset repository. So we have prepared datasets. The first phase of the dataset search and ingest is, as you can see, is an AI-based dataset search. When we look for usually public experiments in the public repository, but it can also be customer-based repositories. But these public repositories are mostly based on repositories like Geo and SRA. And the AI-based search is based on specific characteristics which we search for. The next phase is split into two parallel steps. The molecular data ingest, which processes raw molecular data and saves to lakeFS. And this step we'll be talking about in the next slide. the sample metadata standardization, where we pull the experiment metadata, which is many different unstructured formats, and use, in this case, we heavily use LLM-based models to curate and annotate samples in this data set. This is all based on the CytoReason ontologies, which is the source of truth for all CytoReason biological and clinical metadata. Since the molecular data and sample metadata are not published in the same time, we can use lakeFS actions mechanism to trigger validation to see that the dataset data is aligned. Just as a quick example, does the molecular data has the same samples as the sample metadata? This is a simple validation that we can do as part of the lakeFS actions. The second part of the model engine on the right side is the model factory, where we basically refer to the standardized data sets which we have in the CytoReason dataset repository, which we choose in the model-specific experimental design. We will talk about the model generation later in this session, and we can move on to the data ingestion, which we mentioned earlier. So for the molecular data processing, we changed our legacy custom pipeline to be based on Nextflow. We started by taking the NF-core-based RNA-seq and SC downstream pipelines as a starting point, as they already had more features than we had previously. And I have to say that we have to enhance them a little bit. So there were a few things that we had to improve and enrich these pipelines. These processes are where I have to say these processes are where Seqera and Nextflow really shines. Since the integration with the known repositories, the different format types, the integration with standard paradigm like spreadsheets, I have to say this really saved us work. And in addition, since these processes are very heavy, these data processing pipelines, we also gained a lot from the Seqera features, such as Wave for managing containerized environments and building on-the-fly containers. In our case, it's we use Conda environments, so we leverage the Conda environments to build on-the-fly containers with Wave. Fusion, which was used with Wave to improve container loading, and also to improve I/O usage when working with cloud storage as the work there. In some cases, when we use the Seqera platform, we needed to have data isolation between pipelines, which run for different data providers' origin data. So for that, we use different workspaces within the Seqera platform, so that each process has its own cloud resources and credentials configuration. And by that, we were able to isolate each provider processing. In addition, as mentioned a little bit before about the Seqera Data Explorer, since we published the data to lakeFS, we were able to explore the lakeFS repository and branches with the Seqera platform. This was all made available courtesy of the Seqera product team, which added an S3 compatible data repository support to the Data Explorer section on the platform. And since lakeFS supports S3 API, we were able to run the pipeline and explore the outputs. And this was all done in the Seqera platform. So this is a simple example of how you can configure a lakeFS connection with S3 compatible storage on the Seqera platform. So obviously you need to set your credentials, you need to put your lakeFS server URI, and you use the older path style S3 compatible URIs, and then when you define the credentials and the configuration, you can add a new data repository, which is basically a lakeFS repository. And as you can see here, you can see the differentiation between the lakeFS UI and how Seqera Platform sees the lakeFS repository. So branches are actually represented as simple folders in the Seqera Platform Data Explorer. Okay, so moving on to more integration between Nextflow and lakeFS. So we talked about publishing version outputs to lakeFS from Nextflow pipelines, but Nextflow at its core doesn't have a seamless integration with lakeFS. So how did we integrate lakeFS to Nextflow? So I will go back a little bit, and I will tell you that first, when I started working with Nextflow, the first thing that impressed me was when you publish your outputs on the local executor to a local path, it doesn't actually copy the file, but generate a symbolic link to the file on the working dir. So having said that, the first thing I thought, wouldn't it be nice if this could also be done with cloud storage? Well, there aren't symbolic links on cloud storage, as you all know. But if you use cloud storage as the working dir or the Nextflow bucket dir, which is the new bucket dir feature, when you publish to lakeFS, you will be able to get the same behavior as links. Since lakeFS is a data catalog service on top of the cloud storage and any cloud storage. So this behavior is available seamlessly through Nextflow, only if you use the NF lakeFS plugin, which we at CytoReason developed. And in addition, by using lakeFS with Nextflow, you are able to get atomic publish operations, as the publishing in Nextflow is done asynchronously. You might have a case where after you publish the data for some processes, you might fail on a different one, which results in a stale or corrupted state of the repository. Instead, the plugin uses workflow events to trigger a commit or revert the published data in the repository. And the NF lakeFS plugin is publicly available in the Nextflow plugin registry, which is an open source and can be imported in any Nextflow pipeline used for any customer, obviously. Okay, moving on. Okay, so here's a little example of how we use the NF lakeFS plugin. So as you can see, you can use lakeFS URIs to read lakeFS-based files into a channel, and in a process you can in a process output you can publish those outputs to lakeFS. And just an example which is shown here, you can see that there's a nice feature on the NF lakeFS plugin. So you can attach metadata to the published outputs on lakeFS. And in this specific scenario that we see here, for each file that was published to lakeFS, we attached the Nextflow workflow metadata and the task metadata as part of the published to lakeFS. and this is attached to those files. Moving on to the model factory. As we saw previously on the right side, so previously we had a legacy pipeline. The legacy model factory pipeline was based on a very large DAG, which was all implemented in Erlang, including the workflow orchestration. The pipeline was using custom distributed engine based on GKE, which was tightly coupled in the code. The disease model was published through BigQuery. So in addition to having limited ability to version the disease model, we had data transformation processes to align the R objects with the schema. It also had a good caching mechanism, actually, similar to what we have in Nextflow. And you can only run the pipeline end-to-end for any change. And the analysis code, as it was written, was hard to be separated from the code because data was dependent on our objects. So we had to re-engineer the model generation. So we wanted to do a change to better support the E2 paradigm, which we talked about before. So we did it on stages. We first replaced BigQuery with lakeFS and published Parquet files, which were representing the E2 tabular data from the pipeline instead of our objects. So now it was easier to track the data that was published and release versions. The second phase was that we re-implemented the orchestration with Nextflow to completely decouple workflow configuration and workflow code from the evidence generators, including processes configuration that was bundled in code, such as image versions and memory configurations. We also implemented the evidence generator as Nextflow sub-workflows, which can run independently with input from lakeFS. And the move to Nextflow also gave us the ability to use different types of executors and cloud storage and not be bound to GCP, which that easily led us to the final requirement, which drove us to move faster to do the changes. This requirement was the bring your own cloud, the BYOC, which is the ability to run on any cloud and with any executor. Okay, so moving to BYOC, bring your own cloud, and where did the bring your own cloud came from and why? That's the first thing we need to ask. Well, the basic idea is that the customer said, look, we like your models, but we want to also enhance them with our data. And another constraint that they put on us, our data can leave our premise. So this is, from my point of view, actually, this is quite understandable as a system architect. The customer has many different rules and regulations for protecting its data, since it is a very important asset, especially for big pharma companies. Luckily, this was aligned with the transformation process that we planned for the model factory. So we now had the ability to run the model factory and generate an enhanced model on the customer cloud environment. So Digging deeper into bring your own cloud, this is how it actually works. I'll show it with this animation. So we start with a release model on CytoReason. You can see on the bottom of the slide, we start with a release model on a CytoReason environment on lakeFS. Let's say it's version 1.0.0. And on the customer environment, we already provisioned and deployed new components like the model importer, which pulls the release models. Obviously, we have an on-premise lakeFS installation, and we also have a component called the model enhancement, which is cloud provider agnostic, and it is built out of subcomponents of the model engine, which it's basically model enhancement is a subset of the model engine, which enables the customer to do the model enhancement. So the next stage is basically when we start a project on whether it's a project or the customer wants to use our platform, we do a copy. We actually pull from the CytoReason environment lakeFS. We pull released models, tagged release models. We don't push anything to customer environment. We only pull the data. And those release models are pushed into lakeFS to the on-premise lakeFS, which actually creates a mirroring of the CytoReason environment lakeFS with the relevant models that we want the customer to use with his enhanced model. And once we have the release model on the customer environment, we can use the customer data on the customer premise and read from the release model, which are on the customer environment, on his own VPC environment, isolated network environment. And with that, we can generate with the model enhancement component an enhanced model with a new tag, which we can publish directly on the same repository to the lakeFS on-premise installation. So now we have the previous public model and we have an enhanced model on the same repository, which are represented with two different tags on the lakeFS repository, like a release that you do, exactly like a release that you do on GitHub. Now I want to introduce two scenarios where we leveraged lakeFS versioning to create commits, branching, and tagging. So the first scenario is the model engine scenario of generating a new model or enhancing an existing one. It can also be generating a completely new model. So we start from a branch, which either can be an empty branch if we create a new model, or it can be a previous release of the model. And once we start running the pipeline for generating a new model, we run a specific evidence generator, which basically creates a new branch on lakeFS and a new commit. And through lakeFS actions, which is similar to GitHub actions, we can leverage lakeFS actions to automate data validations on each commit. If we create another commit from a different evidence generator, or it could be that we changed the experimental design and reran the same evidence generator, but we did a new commit, and through lakeFS, the new commit can be compared to the previous commit. And now if we can continue with another commit and another evidence generator, as you can see, it's exactly similar to generating a software-based product. And in this case, it's a data product, which we generate and we finalize it with the final commit, which is an E2 model. And in that stage, we can say that this is a complete model, and we can merge it back to master. And in that step, before we release the model, we can have biologists and bioinformaticians do biological validation of results, for example, with biological expectations. Once this is validated, we can release the new disease model as a new with the new lakeFS tag, and internally we use semantic versioning to version the disease model. The second scenario that I wanted to talk about is more common on the customer project, where the customer adds custom evidences to the model and processes them. So we obviously start from a release model and we work on the customer premise, and we want to add custom evidences. So obviously from the released tag, we can generate a new branch, which is the enhancement branch on the customer premise. And we can add at this point custom evidences, which is a project-specific, specific data or customer-specific custom evidences. And at this point, we can run an isolated evidence generator, which is based on a Nextflow sub-workflow to run specific specific analysis on the newly custom evidences that we added in the previous stage. And once we're done with that, we can release to the customer, or the customer can release by himself, a new enhanced model with the new custom evidences, which are customer-specific evidences that we added to the disease model. Okay, so summing up, next steps that we want to take with the model engine from here. First of all, we want to obviously finish the migration. We want to port the remaining monolith analyses to the independent evidence generators. We want to broaden bring your own cloud with more cloud executors. We want to integrate Lina Research Agent with the model enhancement, and this means that the isolated Nextflow sub-workflows, which are evidence generator, will be able to be triggered from the Lina Research Agent. And we want self-serve composability to customers assemble and enhance models from release version E2 models. Okay, so I'm done here. Thank you all.